Hi, I'm Marina Garrett. I'm an investigator in the MindScope program at the Allen Institute for Brain Science, and I'm going to tell you about a data set that we've generated where we trained mice to perform a visually guided behavioral task and then measured neural activity in different excitatory and inhibitory neuron types with in vivo 2-photon calcium imaging. The task that we've used for this data set is a visual change detection task where mice are presented with a continuous stream of flash stimuli. Each stimulus presentation lasts 250 milliseconds with a 500 millisecond interstimulus interval. And the mice learn to report changes in image identity by emitting a lick to earn a water reward. Once mice are well trained in this task, they transition to the 2-photon imaging portion of the experiment, where we image the same population of cells over multiple days as we vary the sensory and behavioral context that the animal is in, so that we can follow the evolution of activity over time under these different conditions. Mice initially perform the task with the same image set that they were trained on, which has become highly familiar over time, and that's indicated in shades of red here over multiple sessions. Then they are exposed to a novel image set that they have not seen before being under the microscope, and that's here indicated in shades of blue. Having multiple sessions with this novel image set is useful so that we can look at how neural activity changes as the stimuli become increasingly familiar. We've also included interleaved passive viewing sessions for both familiar and novel images. And in these passive sessions, the mice have already received their daily water, so they are satiated, and the stimulus is played in open loop mode with the lick spout retracted so that rewards are not available. And this allows investigation of the impact of task engagement and motivation on neural activity. Another important aspect of our experimental design takes advantage of the fact that mice experience this highly regular cadence of repeated stimulus presentations throughout their training. And during the 2-photon imaging sessions, once mice are well trained and have lots of experience with this regular stimulus timing, we randomly omit 5% of all non-change stimulus presentations. And this unexpected interruption to the regular stimulus cadence appears as an extended gray period to the mouse and can be considered as a violation of temporal expectation. So with this data set, some of the many questions you could ask include how variable are neurons from day to day, and how do different types of neurons respond to stimulus novelty or stimulus omission. So we've collected data with this experimental design using both traditional single-plane 2-photon imaging as well as multi-plane imaging, where we image 4 cortical depths across 2 visual areas in the same session. For this data set, we've imaged in V1 and LM. And we've performed these recordings in transgenic mice that express a genetically encoded calcium indicator in either excitatory neurons, SST inhibitory neurons, or VIP inhibitory neurons. And in total, we've recorded from 82 mice over 551 imaging sessions for a total of over 34,000 neurons, and all of this data is openly available to you for use in your projects. In each session, we measure several aspects of the animal's behavior, as shown here in the top row, with running speed in green, pupil diameter in purple, licking behavior as red tick marks, and reward times as blue dots. The colored bars behind these time series show the timing of the repeated stimulus presentations, with the color of the bar indicating the identity of the stimulus. You can see that for some image changes where the color of the bars transitions, the mouse slows down and licks to earn a reward, while other changes are missed. You'll also note that these gaps in stimulus presentations where stimuli are omitted. Then finally, here in the bottom row is a representative delta F over F trace, change in fluorescence over baseline from one neuron that appears to be selective for one of the images, represented by the pink bars. So by relating these signals to each other, you can ask questions like, how is the activity of different cell types related to behavior? And you can further investigate whether that relationship depends on stimulus or behavioral context, like novelty and task engagement. So we've provided you with 2 ways of accessing the dataset. The first is using the Allen SDK, which stands for Software Developer Kit. And this is a Python-based toolkit that facilitates download and interaction with the full dataset. And the advantage of this method of data access is that it is very flexible. You can access the entire dataset and focus on any aspect of the data that you want, including the full behavioral training history of each mouse, as well as the 2-photon imaging data I've just described. The disadvantage is that it's very flexible, which means you need to be familiar with the experimental design and make decisions about what data you want to use and know how to find it. We have provided an example notebook that shows you how to load data using the SDK, extract relevant information, and reorganize the data into a format that is useful for your analysis. We also have a variety of example notebooks linked here through the SDK Read the Docs page, and I encourage you to skim through these notebooks to get a sense of what is available to you. So we recognize that not everyone may want this level of flexibility or complexity, so we have also provided an example notebook that loads a pre-selected subset of the data that allows you to get started more quickly. And advantages of this method of data access are that it is much simpler, it's super easy to load, data is in a ready-to-use format, so you can get started right away. The disadvantage is that it only includes a subset of the data, so that the range of questions that can be addressed is more constrained. And so this notebook described here shows you how to download the file, explore what it contains, and make some basic plots of activity under different conditions. One suggestion for deciding which method of data access is best for you is to start with this simple notebook, explore what's available with the pre-processed file, and then decide whether you want to move on to the other notebook and use the SDK to access a broader range of data. So if you have questions, you can find a lot of additional information on the main Allen SDK landing page at this top link here. I've also created a kind of cheat sheet slide as a quick reference about the different dimensions of the data set that you might want to explore. And we also have a guide to finding all the data you want using the SDK, as well as a technical white paper that describes the experimental methods in detail. And if you have questions that aren't answered by any of the available documentation, you can post a question to the Allen Institute user forum, and if all else fails, you're welcome to just email me directly. So thank you very much for listening, and I hope you have fun working with these data sets.